Hello, and welcome back to Plantopia, the plant health podcast produced by the American Phytopathological Society. I'm Jim Bradeen, a professor of plant pathology and associate vice president at Colorado State University. And today I'm excited to introduce Dr. Hanareia Ehau-Taumaunu. Hanareia completed her undergraduate studies at the University of Auckland in New Zealand, where she studied sciences and arts as a double major in biological sciences, Maori studies, and writing studies. As an undergraduate student, summer research experiences at the New Zealand Institute for Plant and Food Research fueled her interests in agriculture and plant health. And following her master's studies in biological sciences, Hanareia joined Pennsylvania State University, where she recently earned her PhD in plant pathology. Hanareia's PhD studies were partially funded by a Fulbright Fellowship, and she has been the recipient of numerous awards and honors. And today we're going to discuss her work in collaboration with her graduate mentor, Professor Kevin Hockett. And we'll also discuss her perspectives on Indigenous ways of knowing and her perspectives as an Indigenous woman and a scientist. Hanareia, welcome to Plantopia.
Thanks, Jim. It's great to be here on the podcast. I would like to first acknowledge that I am speaking to you from the lands of the Shawnee and Susquehannock nations that have been present in what we currently call the state as Pennsylvania since millennia. And I just really want to give my sincere acknowledgement to those people, to the land, to the waters that have allowed me to be here for the last 5 years. And that I really do strive to emulate their sustained commitment to their environments and to everything in and around that's around us. So
let me start by saying congratulations to you on completing your PhD just in the last couple of weeks, and I think big changes are ahead for you. We'll talk about that in just a moment.
Thanks. It's been a long journey. But it is great to be at this milestone, and as you said, I'm very much looking forward to the future as well.
That's fantastic. Well, let's start really with plant pathology and your interest in plant pathology. You shared a moment ago that growing up, you were very connected with nature and agriculture, and that's really fueled a lifelong interest. Could you tell us a bit more about your background?
Sure. Yeah, I grew up in a very rural, remote area of Aotearoa, New Zealand, on the lands of my ancestors. And I was I guess at the time I didn't realize it, but I was surrounded by lots of farming. My family always had a very large garden. We were growing kumara, our sweet potato lineage in New Zealand, that I absolutely love. And, just other types, beef farming and all that kind of thing. And it somehow followed me to university. When I got there, I was really interested in biology, and then found kind of plant pathology through my time at Plant and Food, and I was able to connect that with the environment that I was living in and was brought up in, and then I could connect that to the hard sciences that I was learning throughout my undergrad studies, and then also at my time at Plant and Food. So yeah, I remember being asked in interviews, at the end of my undergrad, and they're like, why did you do this? And I'm like, it was just all around me all the time. Why wouldn't I? It was just always that constant connection to my environment, and that I was, taught by different people of how we can work with the environment, where we should grow things, when we should grow things, just as a kid growing up. And that just continued throughout my studies, which was great.
So tell me more about your time with Plant and Food. How did that shape your career path?
It heavily shaped me. It definitely made me the plant pathologist that I am today. I was brought in on a project that was working with Maori kiwi fruit growers and how they were trying to work through the PSA endemic that was happening in New Zealand. So we had a pathogen affect one of our main kiwi fruit cultivars. The pathogen was the bacterium Pseudomonas syringae pathoviractinidae, and it was devastating our kiwifruit growers. And a large portion of our kiwifruit growers are Māori growers. And so they wanted to connect the two. And that's where I came in as a young Māori scientist at that time, taking an interest in plant pathology and plant sciences. And yeah, I got to work with different kiwifruit growers. And then I was doing a lot of PCR on different genes that we were trying to figure out what were they really doing, what were they playing a part in terms of the pathogenicity of the bacterium that we were working with. And yeah, it just really caught my interest, and I could see the connections between plant health, how that affects the communities that I was living in and I'm from, and that I could actually join the two passions of being able to do science that was cool and fun and interesting, that would have a positive impact on my communities and also just a country as a whole.
And plant pathology is definitely a cool, fun science that has a lot of impact. So I think a lot of us could relate to your perspectives there. How did you end up at Penn State?
So I spent a bit of time doing plant pathology in my undergrad, which was wonderful. I spent many summers at Plant and Food, but then also was taking classes as well in those types of areas, just in ecology and also microbial ecology and also all the molecular stuff as well. Then I kind of took a detour for my master's, and I actually went into entomology and looking at the microbiomes of a native insect of New Zealand that had become a pest in agricultural pastures. So it was the enemy of a lot of farmers at the time because it was eating all their grass, but really they were just overtaking the home of that insect in the first place. And so I was able to come in on that project. So I went to entomology, realized that I had such a huge passion for plant pathology and really, really wanted to come back to it. So decided to pursue a PhD in plant pathology. And I was very fortunate to receive a Fulbright scholarship that kind of made me think about doing science outside of Aotearoa, New Zealand, kind of broadening my horizons even more than what it was happening at the time. And so I found myself coming to Penn State to do my PhD and to learn from the people at the university, the science community, apiarist community, but also connecting with the Indigenous communities of this land as well. So yeah, I've been on a winding kind of journey to get to where I am today. But each of those perspectives and learnings and tools that I've gathered along the way have been really beneficial, and I'm very fortunate to have received those along the way as well. So my journey is not over just yet, but yeah, coming across the Pacific Ocean, across Te Moana Nui a Kiwa, has been one of the best times of my kind of a shortish life so far.
Well, we're delighted that you've continued your studies in plant pathology. Just a disclaimer: we love our friends in entomology as well.
Yes.
But we are very happy to count you amongst our community. Now, your PhD work at Penn State also was on Pseudomonas syringae, so a different pathovar, and you have published an incredibly cool manuscript very recently. This actually is currently it's in APS Phytobiome first look, so it'll be published very soon. Probably by the time this podcast is out, it will be published, and we'll make sure that we have a link to this on plantopiapodcast.org. The title of the study is Pathogene Phyllosphere Microbial Communities Develop Suppression Towards Bacterial Speck Disease in Tomato. There's a lot to unpack there. What really struck me is the way you framed your approach. Fundamentally, this is about biocontrol. And you talk a bit about suppressive soils as an intellectual framework for thinking about how biocontrol can work in complex agricultural environments. And then you posit that the same dynamic that happens in that soil community may actually be happening in the phyllosphere and may be something that we could leverage for crop disease management. I guess let's start with phyllosphere. What is the phyllosphere, and why do we care about it?
Phyllosphere is all the microbes that live on the surface organs of a plant. So anything that's on the surface of the aerial part of the plant that can host microbes, we're really interested in terms of the phyllosphere. So that can include, the leaves, the stems, flowers, everything that's above the ground, which is a very unique environment, especially when we compare that to the soil, where, the plant typically in agriculture is there for a very short period of time, and kind of gives us the fruits that we want in many of the vegetables, including the one that I was working with, tomato. And it just, it's such a unique environment because it's so much more hostile. It changes all the time. It's heavily affected by the climate, even the microclimate in amongst those plants. And it's just an interesting space to be in, and trying to translate some of the knowledge that we've gained, say, from the soils, and bringing that up to the phyllosphere.
So your study really looks at that phyllosphere composition, which you characterize as being really dynamic. And yet, at one level, you're assuming that manipulating that environment may actually be good for plant health. Where did this line of thinking come from? What was the impetus for this project?
I think both Kevin, my doctoral advisor, we've both had an interest in biological controls, but kind of expanding the traditional kind of look that we've had. Usually we're looking at one particular microbe that can target our pathogen of interest. And, sometimes, a lot of the times, we're seeing different efficacy results when it's in the field. Sometimes it works in the lab, sometimes it doesn't work in the field, vice versa. And, we will, had some, interest and also looking at other research that was being done in soils, where really it's the whole community that's being involved in a lot of that disease suppression. And so just trying to think about, well, the phyllosphere holds and hosts a range of microbes in their own communities, and how could we change kind of that normal paradigm where we're using a one-to-one system to more of a whole community, to tens, to hundreds, to thousands, to that one. So then it's able to, hopefully adapt during that time that it's interacting with that pathogen. It's also adding in more dynamic and different mechanisms of targeting that microorganism of interest as well. And so it just gives us a bit more flexibility, a bit more breadth in how we can kind of target and really suppress these pathogens that, we want to help the farmers be able to get around and to be able to, have crop production. And then also that this could also be used in natural systems as well, because there are tons and tons of microbes in those systems as well. So it's really thinking about it at the ecosystem level of, okay, there's going to be microbial communities wherever they may be, and how can we leverage those communities to work against our pathogens that we're, trying to help out our world with.
It's really neat. When I first read your paper, my first thought was about the experimental approach. How could you actually track change over microbial generations? Tell us a bit about what you did in this paper.
So we came up with an experimental method where, we had looked at previous research, and that we found that researchers were able to transfer microbes from one phyllosphere to another. So we're like, okay, we can definitely manipulate that ourselves experimentally, where we can take a community from one to the other, and that they would stay relatively the same. That was based on previous research. So we kind of used the passaging kind of experiment where we could transfer from phyllosphere to phyllosphere, thinking about it that it would be a continual crop rotation. And that when we're thinking about suppressive soils, those microorganisms are always present in those soils, regardless of what's been planted throughout the different seasons. And so we knew there had to be continual contact between the microbial community, the pathogen of interest, and then our host. And so for us to be able to do that, but in the phyllosphere, when, tomatoes only last so long, and then they're either removed from the field or they die and senesce and all that thing. So we were taking a method that had been previously done by some other researchers, and that we would constantly cycle, transfer these microbial communities, and then manipulate them through that process. And so, we were transferring these microbial communities. We were introducing our pathogen of interest, trying to, see if we could push the continuum of disease upwards and see an increase in disease, in the hopes that if they were having these continual interactions, again, between the community, the pathogen, and the host, we could potentially see similar dynamics that are shown and have been shown in some of the suppressive soils examples, where we get this huge disease outbreaks, and then there's a sudden loss of that disease severity. And so we designed this experiment in the hopes that we would see this, and fortunately we were actually able to see similar dynamics in our work as well in the phyllosphere. So it was a very high risk experiment for a PhD student to be following, because we were not too sure of the outcomes of this kind of work. But transferring and relating to the knowledge that's already out there in plant pathology really helped us in designing this experiment, thinking about, how is this going to ecologically impact these communities while it's interacting with our pathogen of interest. And it just kind of grew from there. And then I spent many, many months in the greenhouse trying to get this to work. And at least, we were able to fortunately show this particular dynamic, which we see in soils, and now we could kind of experimentally show it happening in the phyllosphere.
It's a great study, and I think your approach really is creative. I would describe it as elegant. At first I understood the general approach that you were taking, but I was really at a loss at how you would do this experimentally. But when I read your paper and understood your approach, I thought, yes, that really makes a lot of sense. And to me, that's elegant science. When somebody else does it and you say, yes, that's of course the way it should be done. So congratulations on this study. I'm curious though, what comes next here, and what do you see as the application for disease management in the field?
As this study is really the first of its kind, this is setting the stage, really. It's a proof of concept that, we could potentially manipulate these particular communities in the phyllosphere for plant health, to work against plant diseases. There's still a lot to figure out in terms of what's actually happening here, and in suppressive soils, that type of research has been going on for decades, and they're still finding new things. They're still finding out interactions that might be important and how we can manipulate those soils. So, this is just the very beginning of this work. I am also currently putting a paper together to show the microbiome side of what was happening throughout this experiment. So, we can see who's there, what they might be doing, and when they're doing it. So we're trying to target, see if there's any key players, any key fungi or bacteria or viruses that might be influencing the disease dynamics in our work. And then also looking at some of their functionalities and what maybe they have a toxin, maybe they have a peptide, who knows what might be happening. So we'll be trying to publish that sometime soon, and amongst everything else that's happening. But really, I think this is really just the beginning of this story. And there are tons of plant pathologists, microbiome people, plant health, and in general are really looking at how we can shift to either biological controls or just using what nature has given us in the first place to work with it, rather than introducing things that aren't necessarily good for our environment at the same time. There's already a student in my lab group that's already following on with this, but with a different pathogen. So, we're trying to see if this can be used for multiple pathogens. And yeah, I think the next thing is trying to think of, if we can implement this at a field scale. It was easy enough to do in the greenhouse. I can control it. I can, manipulate every single part of that experiment. But what really needs to be done is, can we transfer this knowledge to the field? Because that's where it needs to be applied. So that'll probably be future stages of this work. And yeah, it really just comes into thinking about how can we holistically work with our environment to prevent plant disease. And this is one way, because there are plenty of microbes in our environment. that are going to either be beneficial or not at different times. So yeah, I'm very excited for this line of research, and I'm really looking forward to seeing where it goes in the future.
Yeah, so my scientific cliffhanger, stay tuned. We do look forward to reading this paper, but also the future work that's going to come out. Really is a pretty exciting line of research, and I highly recommend this paper to anyone who's listening. So this really wraps up your PhD work. Again, you very recently and very successfully defended. Congratulations. Where does your career go next?
So I'll be heading home back to Aotearoa, New Zealand, to continue my research career as a postdoctoral fellow with the Bioprotection Aotearoa. And I'll also be hosted by Plant and Food Research, so kind of going back to my roots, which is pretty amazing as an early career researcher to be having that opportunity. And so not exactly too sure what I'll be doing throughout my postdoc. That's for me to figure out once I'm back home. But overall, it's really looking at trying to promote healthy ecosystems, both in agriculture and also in our natural systems, as we have many significant plants that are culturally relevant to Māori back in New Zealand. So, anything to do with that. So hopefully I'll be looking at plant diseases and microbiomes in some form or whatnot, but also being able to connect back to Indigenous science, my Indigenous knowledge, and kind of working on research for Māori, with Māori, and really answering the questions that they want answered, or that needs to be answered, so that as a people they can flourish, but also as a country and an environment it can flourish as well. So yeah, I'm really looking forward to heading home. I've spent enough time here in Turtle Island in the USA. And I'll be taking everything that I've learned and sharing it with everyone back home.
That's great. And we wish you the best of luck in this future postdoc. And you mentioned something I'm really eager to know more about, your Indigenous upbringing and how this has influenced your perspectives as a scientist. And this is an area that you've really had pretty significant impact as a graduate student. Recently, I viewed a presentation that you did as part of the Phipps Conservatory and Botanical Gardens Nature of Place symposium that was held just pre-pandemic in February of 2020. I should mention the Phipps Conservatory is in Pittsburgh, and in this talk, which was called To Hononga, the Interconnection of Maori and Nature, you talk about Indigenous perspectives around nature and historical knowledge about the environment and how this has informed your sciences. And you introduced the concept of whakapapa. Could you tell us about what whakapapa is and how it's influenced the way you see plant pathology?
So for Māori who are Indigenous to Aotearoa, New Zealand, whakapapa can be loosely translated in English as genealogy, but it's a bit more than that in terms of, if you think of just the English word of genealogy, whakapapa goes beyond that. And typically when one introduces themselves, they will kind of talk about their whakapapa, and that will start with the place and the area that they're from. So you're connecting yourself to the rivers, to the mountains, to the oceans. This is you, and that's your whakapapa. Yes, you have your human whakapapa, so you have your ancestors that have been on those lands, in those rivers, in the oceans that you kind of come from. And so your connection is more than just you and your immediate family. It's really you, your family, your community, your larger community, which also includes the environment. Everything is connected, as in the title with Tu Honotanga, where it's the interaction and interconnectedness of everything that is around you. But you are not central to that either. You are just part of everything that is around you as well. So for whakapapa and thinking about our place in nature and also plant pathology, really what I'm trying to do as a researcher, but then also just as a person who is belonging to a particular area. It's taking care of everyone and everything in that environment that I'm in. Because I whakapapa to them, and so I should be also a caretaker of that because they are a caretaker of me. And so it's really holistic, kind of cyclical in nature, in how I've gone about learning Western science and the tools that we have in Western science, and adding that to the science that we have in my cultural knowledge, in my traditional knowledge that I've learned throughout my being, and really bringing the two together, because now that I think about it, that is my whakapapa. Yes, I have my culture, but then I've also learned all these other things from other people who come from other cultures, and now that is part of me. And so, yeah, as a researcher, that's really my research whakapapa. In a sense, that's my research genealogy, and that's what I'm hoping to strive to do when I head back home.
So tell me more about your time in the US and how your Indigenous perspectives have created opportunities or maybe challenges for you throughout your grad school period. How have people reacted?
So I think for myself, yes, I'm an Indigenous person, but I'm a visitor to these lands. You know, I'm currently staying and occupying on the lands of the Shawnee and Susquehannock nations, and so yes, I'm Indigenous, but I'm not Indigenous to here where I'm currently located. And so it has been a wonderful gift to be able to learn from the peoples of these lands, and be able to visit them, talk with them, share stories, find connections in between our cultures, you know, how we think about the world, how we want the world to be, and being able to share experiences, you know, the good and the bad, you know, thinking about colonization, how institutions are, you know, aren't necessarily indigenizing themselves or even welcoming of Indigenous peoples, you know, and then to going and learning from, you know, the elders of different nations that are sharing stories that have been passed on for millennia and things like that, and combining that in my time here in the land that we currently call the United States of America. So it's been interesting, but then I've also been asked to be the Indigenous person at things, and it's very uncomfortable because I'm like, this is not where I'm from. I shouldn't be the person doing this, but I'm the only one here. So, you know, I've been as supportive as I can of, you know, finding community here with other Indigenous peoples from around the world. I think that's one of the greatest things that university institutions kind of have, is that they bring a lot of people together from many, many different places. And so I've been able to connect with Indigenous peoples from all over the world just being at my university here. So yeah, there's positives and negatives, and there's been challenges in terms of being an Indigenous person. I am also a white passing Indigenous person as well. So I can't necessarily talk to the struggles of those who look more like what people assume they would look like. I can kind of glide in and out, but once they hear my name or I start talking, you know, very in a way that I speak naturally as an Indigenous person about the world, then a lot of people's tunes will change. Yeah, as you know, and also in the scientist world, there's not many Indigenous plant pathologists that I've been able to meet so far. I've met a few, but there is not many. And thinking about, you know, as plant pathologists, we're here to promote plant health, to promote healthy ecosystems. And there really shouldn't be a clash between the two, because as an Indigenous person from where I'm from my views at least, those two, that's the same, where I'm thinking about the environment, and so are plant pathologists. You know, we're wanting to make sure that there is an environment for the next generation so that they can grow all the plants that they want, and you know, all over the world. So yeah, it's been an interesting time being away and also being an Indigenous person as well.
And you mentioned having the opportunity to interact with Indigenous people from all over the world, and that really is one of the great things about our university communities, the diversity that uh, these educational institutions foster. What has that been like for you? What are you finding that Indigenous people from throughout the world are very different, very similar? Are there commonalities that you've come across that surprised you?
Yeah, for sure. There are definitely many experiences that we can share that are similar, in terms of the sentiment, the emotion that goes behind it. Also, just some of the great epistemologies and different stories that we have, how we kind of connect with our environments. Either many of us will, use a stellar lunar ecological calendar, so it is place-based kind of knowledge, but it's all different for where we're from because our environments are different and need to be adapted to. But overall, like, we're using very similar kind of ways of trying to understand our environment. And so that's been great. We can share stories. Many of our ancestors have already interacted many, many hundreds of years ago, whether it's over a boat and now it's over an airplane. So, there is many goings and backwards and forwards between many of the people that I've met in terms of culture-wise, which has been amazing, because as an Indigenous person, the more perspectives you have of the world, of what people, need in their things, then you can appreciate what you have back at home, but then you can also help everyone improve together. Because we, as Indigenous people, it's all about the communities, and that communities is not just the humans, but all the communities that are around us, which I guess in our sense includes the microbial community, which is pretty cool as well. So yeah, no, I've been able to meet a lot of different people. The one thing that I have enjoyed, well, one of the many things that I have enjoyed while being here on Turtle Island, is that I arrived and found out that they also make fry bread here, which I didn't know when I was coming here, and I thought I'd have to leave it back home in Aotearoa. I was like, oh no, I'm going to miss out on my fried bread. Got here, they're like, we've got you. Here's some fried bread, enjoy. And so I've learned many, many different ways to eat fried bread now, that I can now go home and kind of share and kind of experience with my own family when I go back there. So yeah, there's all sorts of connections, including Indigenous science as well, just thinking about how we think about the world and how all the different observations, have been made for millennia, that is now kind of seeping in to the traditional Western science world, or at least they're finding out stuff that we knew for ages, and how we can kind of work together to benefit our environments and our communities as well. So yeah, no, it's definitely been a highlight of my time being here. It's connecting with all the different communities that I've been able to connect with.
It's wonderful, and to know that you're taking those perspectives back to New Zealand as well, and incorporating into your science. I'm curious though. We're obviously living in a time of really significant climate change globally, and I for one always tout science, and of course I mean Western science when I say that, as part of the solution. Where do you see Indigenous perspectives and Indigenous science fitting into climate change?
I think Indigenous people have been at the forefront of the climate change kind of issues and solutions since it probably became a term, or even before then. As I said, at least for me and my experiences and my culture, the environment is at the forefront of everything that we do. And so if climate change is affecting that, then obviously we would take action and find solutions to support that, so that for us, so it's the next 3-4 generations are still going to have the pristine environment that we've enjoyed while we've been here on this planet. So I, at least from my knowledge, they've always been there. They've always wanted to be part of the conversation that is being taken without them. And there's many UN delegations from many, many different tribes across the world that have gone there to talk about how the effects of climate change are really heavily affecting them, particularly seeing as about 80% of our diversity is taken care of by Indigenous communities, Indigenous peoples, and how they're trying to take care of those, but what else we're doing in the world is making that harder for them. And we want to keep that biodiversity, and that's not just in plants, that's in animals, microbes, and everything in between. So in terms of climate change, at least from where I'm from, they've always been a part of that conversation, always concerned about the environment and the toll that it's taking on our environment. I think it's just that we have been heavily excluded from communications that might be, or talks that are happening at that higher kind of governmental, kind of worldwide conversation. And I think it's about time that the tables were kind of turned. And, if we put all Indigenous peoples in charge of climate change, I think we could really, really start making moves to kind of, protect what we've got before it's gone.
Yeah, I want to thank you for widening my perspectives on our science and making science more inclusive. There's a lot to learn, and I appreciate you really being at the forefront of talking about Indigenous science and its intersection with plant pathology in particular. We really do need your perspectives, and I appreciate that. I'm wondering if you have any advice for, I don't know, graduate student mentors or employers who are maybe in a position to support Indigenous scientists early in their career. What do we need to know?
I think firstly, I don't think it's necessarily has to start at the individual level. It'll be great if it does, but obviously systemic change would be amazing. Thinking about decolonization, indigenization of our institutions that hold many of the faculty that as students we have to work with to get our degrees and things like that. But it'd be great to see more Indigenous professors in science and plant pathology. So to be able to see ourselves in a system that wasn't built for us would be great to start off with. And then I think it's really just supporting in whatever avenue that student wants to take. And I think we've done that partly in plant pathology. We're always thinking about, the farmers, the growers, what we call stakeholders, and making sure that what we're doing is having an impact for them. So now I think people just might need to broaden that aspect to be like, okay, how is this affecting this community, or in particular this Indigenous community? And just because they're not where you might be working now because of colonization and all the impacts that has happened since then, that you should still be trying to reconnect with those people, that the lands that you're on were traditionally theirs and were taken care of by those people. And so it's really just broadening that horizon and trying to take, an interest and do your own readings and listening to different podcasts and things like that are around Indigenous science, because there is plenty of them and I love them to bits, because you can learn, science is part of the ways of knowing of different cultures. And so what I like to say about myself is that I'm a learner of sciences and not science, because there are many different sciences in the world that can be used and weaved together to really come up with some, creative and innovative solutions that we always talk about and we always want to see. And I think that is definitely the next frontier, is that if scientists who are not exposed or traditionally around Indigenous communities can really make some connections and support the people and what their questions are. As I was saying before while we were chatting, I want to do science for Māori, by Māori, with Māori. Because in the end, I am not necessarily an agent of change, but I have all these tools that I can help support other people in their journeys and what they want to do in plant pathology and trying to prevent plant diseases, but then also trying to create those healthy ecosystems that we all want, really. I think Indigenous science is the way to go. And I think the more you learn, the more you expose yourself to. If you're not in these kind of areas or circles, then I think you'll only see a benefit to yourself, to your lab group, to the universities, to the institutions, to wherever you are. I implore you and implore everyone to really, really make an effort, even just to learn whose land you're on, what the people were, how they were, what they are now, as many of them are still here today, and then connecting that with your science and being like, this is another community that I can work with and support, including our students. I had a great advisor in terms of him being supportive of really doing whatever I wanted in terms of connecting with Indigenous communities, going to conferences like SACNAS and ACES, who are all about, diversity in science, Indigenous scientists from this area, and then just being able to really connect and learn from the people here about their struggles and also their wonderful and amazing cultures as well.
That is incredibly inspiring. Thank you so much for your perspectives and for taking time to meet with us on Plantopia.
You're welcome. It's been great talking with you, Jim.
We just heard from Dr. Hanareia Ehau-Taumaunu, who recently completed her PhD at Pennsylvania State University and will be pursuing a postdoc with New Zealand Institute for Plant and Food Research. I'm Jim Bradeen, the host of Plantopia podcast. Thank you so much for joining us, and we will talk next time.